Yeah, I don't think it'll be hard.
Oh, yeah. It's like somebody that's.
I was like, right now.
Oh, facts. Right. Yeah. I did the right. And so I got paired up. I was the last cohort, but I got paired up.
Oh, nice.
Yeah.
Was that like surgery? No, it's no, I'm telling you, I saw
Yes.
I would answer confidently.
Okay, we will, in the problem set, apply some of those ideas of gel electrophoresis to problems. So you'll get to look at how to interpret data from that. So that will be useful. Oh, I have no screen. Okay. There we go. All right. So we are talking about enzymes starting today and spending quite a bit of time on that. All right, so I think first learning about some concepts of enzymes, how to think about how they catalyze reactions, and how that impacts or does not impact free energy. So things to note here are that enzymes are catalysts, powerful catalysts, and they're very specific to their substrate. So they will bind random things in the cell and catalyze that reaction. So they're very specific to what they bind and the type of reaction they catalyze. So obviously, in terms of being a catalyst, they increase the rate of a biochemical reaction, much higher than those reactions would occur if there's no enzyme around. But they do not alter the equilibrium, meaning the equilibrium constant of a reaction, and they don't alter the energetics of a reaction. So the equilibrium constant meaning what was the ratio of product to reaction to reactive that is present at equilibrium? It doesn't change that number. It doesn't change the delta G not product. It doesn't even change a delta G for a reaction. What it will help is equilibrium to reach faster. Okay, so meaning we know that delta G changes is a consequence of concentrations of products and reactant. Delta G changes, and what it basically allows is for delta G to reach zero fast. So enzymes we will often see require cofactors. They might be metal ions. They may be larger groups that are embedded into the enzyme. So we will see that present. And our free energy value for a reaction is just going to, again, always, as it does, tell us whether a reaction is going to proceed without an input of additional energy. The delta G value won't change given a certain set of concentrations, given concentration. Okay, why do we care about enzymes? They are catalyzing most of our reactions in the body, so understanding how they work and how they're controlled is really important to understanding physiology. And this is just an example of, one reason why enzymes are interesting is in the context of this virus, HIV, human immunodeficiency virus. So the virus was studied in great detail, and we know all of the enzymes that the virus encodes that are required for its life cycle. A couple of those being reverse transcriptase. It brings this along because the cell that it infects does not have an enzyme like reverse transcriptase, which makes DNA from RNA. We don't do that in our cell. It also requires a protease for its infectivity. So these these proteins were studied, their structure was found, and people have discovered inhibitors. So these are small molecules here that fit into the active site or potentially other locations on the enzyme that inhibit their activity. So it basically keeps the HIV from infecting cells. And this has completely changed consequences of getting this virus. 40 years ago, that was a death sentence to be infected with this virus, and now people live with it, which is incredible. So that was a consequence of studying all of the aspects of this virus and finding inhibitors for these particular enzymes. The virus tries to evolve to change so that its enzymes no longer get bound with these inhibitors. But what people found is if you give a so-called cocktail of inhibitors, that the virus can't evolve mutations to all of them during one life cycle. So it's a very effective drug regimen. Anyway, so that's an interesting example of why we study enzymes, but there are many, many more. Okay, so let's talk about enzymes, and this is a little theoretical today, as it always is at the beginning, and then we get into more examples. Enzymes are generally proteins. We know that things like RNA can catalyze reactions, but at this stage in evolution, most of our enzyme reactions are catalyzed by proteins. So we'll be focusing on that. And the term where a substrate binds to an enzyme is called its active site. And that's where the specifics of the reaction happen. And so we talk about that complex in terms of an enzyme substrate complex. This would be an example of a free enzyme. This is substrate that is also not bound yet. And then those two come together as a complex. Okay? That is reversible. So that is not a permanent interaction. So an enzyme and a substrate can come back apart, or it can proceed further and make product. And we will talk about the tendency to do one or the other. Okay? The binding process is mediated by non-covalent interactions. So how does a substrate bind the active site of the enzyme? It's all of these interactions that we've been talking about. Hydrogen bonds, bonds between side chains of amino acids, so ionic bonds, dipole, etc. Even hydrophobic interactions. Yeah.
Do you think the lock and key analogy is accurate, or is it more of just like generalized?
Yeah, so we will talk, I think that's next lecture. It's really more of what they call an induced fit model. So lock and key model is like the idea of a substrate binding an active site in a very specific way where all of the shapes match up. And that didn't take into account then how an enzyme helps to form the transition state, which we'll get to. So what seems to happen is when they bind, they both change shape. So that's called induced fit. And yeah, we'll talk about that soon. So the binding is very specific. Like I said, non-covalent interactions typically. Sometimes we will actually see temporary covalent interactions between a substrate and an enzyme, which is interesting. But we do not change, the enzymes don't change the equilibrium constant. And so in turn, they do not change delta G naught prime. Those are values that do not change. Okay? How do they make the reaction go faster then? They are going to lower the activation energy of the reaction. This is a term we have not heard yet. So we'll discuss what is activation energy and where does it come into play when we're calculating delta G, or does it? Okay, so just a few more thoughts on enzymes and their substrates. So they're very specific in the substrates they recognize and also the type of reaction they're catalyzing. So they don't randomly catalyze different reactions. They catalyze a very specific reaction. And this is, I'm going to show you an example of one. a protease is an enzyme that catalyzes the hydrolysis of peptide bonds. So it breaks peptide bonds. Why does this happen? It happens in our digestive system, right? So if you eat protein, in order to absorb it into the small intestine, the peptide bonds have to be broken because we can't, our cells can't take up big protein. They just take up amino acids. So peptide bonds are broken, and then you start getting the components of proteins. There are hundreds of different proteases in the body, and they all catalyze breaking a peptide bond, but the substrate, meaning the sequence of amino acids that they cut between, is different. And it can be specific in terms of their sequence. So this is an example. This is not something you have to memorize, but it's an example of a specific protease called thrombin. This is an enzyme that is important in the blood clotting cascade, which is a sequence of different reactions that happen when you need to have blood clotting. So this one is so specific in that its substrate is a sequence of amino acids on a protein that has this particular sequence, which everybody knows now, right? And it is cutting between the arginine and the glycine, that peptide bond. So that's how specific it is. So it's recognizing essentially a particular protein, and it's narrowing down this particular region, it's cutting this, and that's really important to the blood clotting cascade. So that's how kind of specific those can be. All right, so these are, again, this is a table, you don't have to memorize this table, but this is an introduction to some of the types of classes of enzymes that we're going to be seeing. The first class is called an oxidoreductase. Those are enzymes that catalyze oxidation reduction reactions. So transferring electrons from one molecule to another. In this case, we are oxidizing ethanol and reducing NAD+. Transferases, those enzymes transfer groups from one substrate to the other in the reaction. So in this case, we are transferring a phosphate from ATP onto the glucose. We'll see that in the first step of glycolysis. That's the first reaction. So that's a transfer reaction. Hydrolase, not surprisingly, adds a water and breaks a bond. So our peptidases would fall under that class. Lyases are groups that decarboxylate, so they break a carbon-carbon bond. In this case, we're releasing a carbon dioxide. And that's sort of the opposite of a ligase. A ligase is going to make a carbon-carbon bond, so ligation there. And then an isomerase is an enzyme that catalyzes just a change in form of the substrate. So we're not changing the atomic composition there, we're just changing its structure, essentially. Yeah.
So is the only difference between a ligase and a transferase that a ligase makes a carbon-carbon bond specifically?
Yeah, exactly. So the transferase is a little more general in that you can all have all different groups that are just transferred from one to the other. And yes, ligase is a carbon-carbon bond. Okay, so those are general classes. We will see examples of all of those as we go through metabolism. So just so you're familiar. Okay, so let's think about chemical reaction rate, how fast enzymes catalyze reactions. And this is a useful way to describe and compare different enzymes, how fast they can catalyze the reaction. And this is often studied, if you have a particular enzyme, like one of the HIV enzymes, and then you add an inhibitor, you want to see how much it's slowed down, right? So you will compare reaction rates between a regular enzyme and one that has an inhibitor. And we'll get into that in a few lectures too. But so let's look at like rates of reaction. What does that mean? We talk about that reaction as velocity. So velocity of the reaction. And we could quantify that in the lab by either measuring disappearance of the substrate over time, meaning if we're going from substrate A to product B, as the reaction proceeds, you'll have a change in A, meaning that will disappear over a certain amount of time, and you will see the appearance of B over time. So both of those describe velocity. So how fast is that? For every A that you use up, you would get one B in this reaction. So that's the rate. So what does the rate relate to? It's going to be described by a value called little k, which is called the rate constant of the reaction. That's just a description of how fast the enzyme is. And sometimes we call that turnover rate, which is basically reactions per second, how fast an enzyme is. So a velocity for a reaction is going to depend on how fast the enzyme is, but it's also going to depend on the concentration of substrate. What would the velocity of the reaction be if we didn't have substrate? It's going to be 0, right? So that's what you can see. Velocity is related to substrate concentration. If you don't have any, velocity is also 0. But it is also impacted by just how fast the enzyme is. Okay, so a slow enzyme, even if you have a lot of substrate, the velocity will be kept lower. Okay, so those are two. And we can kind of see that idea here in terms of concentration of A over time diminishing, and you would also expect to see the same increase in B over time. Okay, everyone kind of understand that idea? Okay, so this is looking just in a forward direction. Reactions can potentially also happen in reverse. So we would also describe the velocity in reverse too. And the reverse rate of reaction is going to be the rate of the reaction in the reverse direction, which they call K-1, the reverse rate. And that velocity will be described by how much B there is. Okay, because we're going the opposite. So in this case, the substrate is B, and the velocity in the reverse direction is K-1 times B. So at equilibrium, you can imagine those are equal to each other. The forward rate times how much A there is going to be exactly equal to the the reaction rate in the reverse direction times how much B there is. That's equal. And we're going back and forth at the exact same velocity. Equilibrium doesn't mean the reaction has stopped. It just means the forward velocity is directly equal to the reverse. Okay. So remember a while ago, we were talking about an equilibrium constant. Equilibrium constant is basically how much product we have at equilibrium over how much react. That's the equilibrium constant. And that is directly related to the delta G naught prime. So those are all things that don't change. So if we rearrange this equation at equilibrium, and we know the equilibrium constant is B over A, we can also describe an equilibrium constant based on reaction rates. So the forward reaction rate over the reverse will also tell us an equilibrium. So, the enzymes are altering the rates of the reaction. The forward rate and the reverse rate with an enzyme is going to be much faster than without an enzyme. So it's going to increase or reduce the time that we need to get to equilibrium. Product is formed much faster. We then reach equilibrium much, much faster than without an enzyme. Sometimes without enzymes, that can be on the order of hours, days, years, thousands of years. So some reactions are essentially not happening unless there's an enzyme. So an enzyme will have an effect on the reaction rate, both in forward and reverse. It will speed up both. It doesn't just speed up one direction. Okay, so if we plugged in some numbers here and we said for this particular reaction going from substrate to product, that the forward rate of reaction is at the rate of 1 × 10⁻⁴ per second. And the reverse rate of reaction is 1 × 10⁻⁶ per second. If we put those into this equilibrium constant equation, that will tell us what our equilibrium constant is. So what does this tell you about at equilibrium for this reaction? What's the relative ratio of product to substrate at equilibrium? Just depending on just calculating the rates of reaction, right? It tells you that there's 100 times more product than substrate at equilibrium. And we can figure that out just by the rates of reaction. Yeah.
Just why, in the bold, it says KF and then times concentration of substrate. But then when you below that, K equilibrium, it's concentration of product over substrate, but then it's equal to KF over KR.
So we're rearranging this and we're dividing by substrate. Yeah, okay, yeah, that's just the rearranging that. And so that is basically telling you that the reverse basically gives you a ratio of the forward rate is going to be most related to the product concentration. Okay, so those are all related. Those are all things that aren't going to change with enzyme concentration. But you can figure out an equilibrium constant using those rates. Okay, now if we have a somewhat more complex chemical reaction where we have more than one substrate, which most of them do, that we end up looking at in metabolism, where let's say we have A plus B goes to C, you can imagine the velocity is going to depend on the concentration of both of those along with the rate in the forward direction. Which makes sense, I think, because if we don't have, let's say we have very little A, but we have a lot of B, then we're limited by how much A we have. So that's going to slow down the reaction. If A is close to 0 in terms of concentration, the velocity is going to be very slow because that's limited. So the velocity is going to be related to both of those concentrations. So most of the enzyme reactions we see will actually be these second order reactions, and they can have multiple steps in the active site, multiple parts of the reaction. And the slowest part is always called the rate limiting step. So we will hear that term. Okay, so let's think about the energetics and relate this to enzymes. And because we have quite a bit of knowledge about delta G now, and we'll see how enzymes don't impact it, but make reaction rates faster. How does that work? Okay, if we have a graph of free energy here, and we know our delta G is the free energy of the products minus the reactants. If we look at A to B here, is the forward direction going from A to B, is that a favorable reaction? Yeah, it's a negative delta G to go from reactant to product in this state. This is at a lower free energy than the reactant. So that delta G in the forward direction is favorable, negative. How about going in reverse? That would be the opposite, the exact amount, right? Going from product back to reactant here, unfavorable by the exact same value in the positive direction, positive sign. Okay, so we know that We feel confident in describing what is favorable, unfavorable. If delta G is 0, the reaction's at equilibrium, and there's no net change in either concentration. So how do enzymes work then? How do they make a reaction faster if they don't change delta G? They don't change the favorability. So this is basically a part of any reaction that is present, but that isn't calculated in the delta G. And this is the activation energy of a reaction. This essentially keeps all favorable reactions from proceeding spontaneously, meaning spontaneously out of control, just proceeding without any stop to them. So a reaction here where our delta G just takes into account the free energy of the products and the reactants. What we don't see is this transition state energy that we have to get over in order to make the product. Is it favorable to go from a reactant to the transition state? No, it's really unfavorable, right? It requires a lot of energy. So what enzymes do is they lower this transition state energy. They lower that activation energy. And we'll talk about how they accomplish that. How do they make that easier? But that's what they're doing. And by lowering that transition state energy relative to an uncatalyzed reaction, it makes the reaction much, much faster. So if you lower that free energy of transition state a little bit, it makes the little k value much, much faster. Do these look familiar to anyone? Or anyone doing OCHEMs or did OCHEM or what have you? Do not panic if you haven't. This is just an example of energy states of a hexose, right? So a sugar, using it as glucose in the context of metabolism, but glucose, let's say, can adopt different forms, and they are at different energy states. It can be in a boat form, it can be in a half chair, it can be in a chair. Chair is the lowest energy form. Okay, so if this sugar wanted to go from a boat to a chair, great, that is favorable, but it has to, along the way, adopt this half chair conformation, which is actually at a higher energy. So that is the activation energy of getting over here. So what an enzyme would do is to sort of stretch it essentially into this unfavorable shape, and then it can adopt the favorable shape. So it makes this process of forming that a little easier and require less energy. Yeah.
To change the shape of the substrate?
Not necessarily. So the question is, does it cost ATP energy for the enzyme to change the shape? So it would or could if the reaction is endergonic. So sometimes it uses ATP if a reaction is endergonic. If a reaction is exergonic, it doesn't require energy, but it uses the favorable interactions, these non-covalent interactions, to help form that transition state. So it makes these temporary bonds that essentially, using the induced fit model, kind of stretch it into a transition state or compress it or whatever, that helps that to form. Yeah, it's not necessarily that it requires ATP. Good question. All right, questions on any of that? Enzyme intro, we will get into more. Go ahead and start problem set. I'm going to talk about one of the questions as a group, question 6, interpretation of that data altogether. So I will get into that in a few minutes.